Welcome to our webinar, USP 922, Water Activity Method, Incorporating Water Activity into Pharmaceutical Safety Programs. My name is Orna, and I'm going to be your host today. A few words about NewTech Group. NewTech Group is a market leader in implementation of innovative technologies in the quality control and research and development laboratory. Our goal is to help you, our customers, improve your laboratory workflow, productivity, and efficiency, and consequently, of course, your results. Our solutions help automate and alleviate many common, repetitive, tedious practices commonly used in every step of the quality control lab, from sample prep through agar pouring and colony counting. And other solutions can be used for analysis of water activity, color, shape, and size. In the realm of water activity measurement instrumentation, we are the North American distribution arm for Novazina Swiss, who has been a world leader in the industry and has been manufacturing equipment solutions for over 50 years. I'd like to introduce our senior application scientist, Dr. Brady Carter, who will be speaking today. Hi, Brady. Dr. Carter specializes in water activity and moisture sorption applications. He earned his PhD and master's degree in food engineering and crop science from the Washington State University and has a bachelor's degree from Weber State University. Dr. Carter has 20 years of experience in research and development, has been the instructor for water activity seminars in over 23 different countries, and has provided on-site water activity training for companies around the world. He's authored over 20 white papers on water activity, moisture sorption, isotherms, and complete moisture analysis. He's participated in hundreds of extension presentations and has given talks at numerous scientific conferences. After the presentation, if you'd like a copy, please just reach out to us. We plan on having time for questions and answers. Don't hesitate to post your questions throughout the presentation, and we're going to do our best to answer. One last thing, at the end of the presentation, we're going to post a link with a short survey in the chat bar. We would really appreciate if you take a few moments to complete it. And on this note, I will transfer the presentation over to Brady. Dr. Carter, take it.
Thank you. Thank you, Orna. Sorry, I was slow on the mute button there, but hello, and hello, everyone, and welcome to our webinar today, USP 922. Obviously, the purpose of this webinar is to discuss the USP 922 new water activity method, recently made official by USP, and to spend a little time talking about what's in there, what you need to know, and then we're going to spend quite a bit of time talking about the applications that are outlined in the method, as ways to be able to take advantage of and utilize water activity for pharmaceutical products. So to start out, let's just do a little bit of a history of water activity and pharmaceuticals. Water activity, of course, has been a key part of regulations for foods for many years. Built right into the regulations as a critical control point because of its ability to provide, primarily to provide microbial safety. In the pharmaceutical side of the or industry, was a little bit slower to adopt the concept of water activity as a way of being able to control the risk of microbial contamination. But back in 2006, USP did publish an informational chapter, USP 1112, on the concept of being able to utilize water activity in non-sterile pharmaceutical dosages as a way of being able to provide some quality and safety assistance. Again, this was an informational chapter, not an actual method, but it did provide some justification for the implementation of water activity testing as a way in a drug release program to provide safety from the chance of being able to have microbial contamination. And so in this USP chapter, it talked about using water activity to optimize product formulations, reduce degradation of active ingredients, reduce its susceptibility to microbial contamination, but perhaps the most useful part of it was this last part of providing a tool for the rationale for reduced frequency of microbial limits testing. And so what that means is that it essentially allowed for, in a way, to justify in a drug release program to limit or reduce the number of microbial limits testing that was done on pharmaceutical products if their water activity was down in a low range, the below 0.7. But a lot of these are solid dosage tablets or similar types of products, very low water activity, much lower than 0.7. So they are not able to support the microbial growth. And so it didn't make a lot of sense to do a lot of testing to try and find microbial contamination on these types of products, as long as good manufacturing practices were being used so that there wasn't the chance of a high level of inoculant coming onto these products. There was really no reason to think or that could be possible that those microorganisms would be able to proliferate on those products at that low of water activity. So it doesn't make sense to do as much testing on those types of products as you would on a product whose water activity is high, where there is the chance and the possibility of that contamination occurring. And one key aspect of this to understand is that in reality, being able to add water activity as a potential control mechanism for, or a risk reduction in terms of microbial contamination is more effective than actually doing microbial count studies, because when you do a microbial study, you're relying on that through random selection, you are able to find an inoculant or a source within a given lot. And you may find it and you may not. There's always a chance that if you didn't sample where the inoculant was, you would never see it in a microbial study. However, with water activity, it being an intrinsic property of the entire lot, so if you measure the water activity, and that's the water activity of the lot, then all of that lot is protected by that low water activity. Again, under the assumption that inoculant is coming from another source, so that you actually have a higher level of safety because of that than if you were to try and determine its safety just through microbial studies without the use of water activity. So USP 1112 published since 2006, but it was not an official method. It was an informational chapter, not an actual method. And so in response to that, USP has been working and is now officially released their new USP water activity method, USP 922. If you take a look at this document and you go through it, what you'll find is there's a section at first on theoretical background, then talks about some factors that affect water activity, talks a little bit about the different sensor technologies that are available for water activity measurement, how to qualify these water activity meters, and then it also includes a section on suggested or recommended applications for water activity. And so what I want to do is sort of follow this outline in terms of what we're going to discuss throughout this webinar. So we're going to basically go through section by section the method, the USP 922 method, and talk in more detail about each of those sections, maybe even provide or hopefully provide some information that's not necessarily contained within the document itself. So to start out, let's talk a little bit about what water activity is. And this is a really important piece of this webinar, because in a lot of cases, when people adopt water activity as a quality test, they do so not fully understanding what water activity is, or possibly even having a misconception of what it is. And so it's important to have an understanding of exactly what water activity tells you in a product. And it's really rooted in the fundamental laws of thermodynamics, where we talk about energy exchange and energy conservation. and that there's a certain amount of energy, chemical potential energy, stored in any kind of interaction that occurs in a particular product. And these interactions could be between different solutes, but as important to our discussion, it's in those interactions between water and other solutes or other water molecules, and how much chemical potential energy is stored in those interactions. They could be ionic interactions, hydrogen bonding interactions. So different types of these different types of interactions. Within those interactions, there is a certain amount of energy, chemical potential energy, stored in those interactions, which means that it's being taken or transferred away from the water molecule. So the water molecule itself will have a certain amount of energy. We can typically measure that through an enthalpy. But when that water molecule interacts with some other material within a product, there's a transfer of energy into that interaction. There's a certain amount of chemical potential energy then stored that came from that interaction and from the energy that was within those molecules themselves. And so in order to look at that and to identify that internal energy of any kind of a given molecule, in this case water, we use something called Gibbs free energy equation. And that's what you see on the screen. This is a derivation of that Gibbs free energy equation that tells us something about how to determine that chemical potential energy. That energy is conserved, it's stored in these interactions or in these products as chemical potential energy. It can be transferred into kinetic energy, but it's that potential energy being stored in those interactions that are key and critical for our understanding of what water activity is. So what Gibbs free energy tells us is that the chemical potential energy of any species, in this case water, is going to be equal to its chemical potential energy in the pure state. So what is the enthalpy of the pure water, or what is that energy of pure water, and then modified by some sort of a factor that's going to change that chemical potential energy from being equal to what it would be in the pure state. So this last part of the equation modifies or changes this value to be some other value less than the chemical potential energy of pure water, which means this actual chemical potential energy of water at any certain state or any when it's inside of a material is going to be some value less than what it is in its pure state. And so the key part of this equation for us is this second part. And in here we have the gas constant, we have the temperature, and then we have this natural log of fugacity. So what this tells us is that because this value is constant, so it's going to be the same for water, this value is a gas constant, so it's constant. If we control temperature, then it remains constant. So the only thing or part of this equation under those conditions that's going to change the chemical potential energy is if the fugacity changes. And so it's this fugacity that we are going to focus in on. And it ends up that this fugacity has another way of describing it, which is the escaping tendency or activity of a species. So in the case of water, we call this water activity. So we take water activity directly from Gibbs free energy equation and show that it is proportional, directly proportional to the chemical potential energy. So as this number changes, so does the chemical potential energy. And this number is changing based upon the ability of water to be able to escape, in this case, to escape in the vapor phase, because of the amount of energy that's associated with its interactions. Those water molecules that are having very tight interactions with other solutes, and there's a lot of energy transfer, they have a much lower energy, and they don't have the enough chemical potential energy to transfer to kinetic energy to be able to escape in the vapor phase. And so there's less fugacity in that case. Now, if the fugacity is the same as it is in the pure state, then this value becomes 1. The natural log of 1 is 0, in which case the chemical potential energy does equal that of the pure substance. So as this number drops due to interactions, due to whatever is actually reducing the escaping tendency of water, and remember, we're not talking on individual water molecules, we're talking overall. So it's an averaging effect of what all the water molecules are doing within the product. So from our description of Gibbs free energy, what we find is that the correct definition for water activity is it's the measure of the energy status of water in a system. It's that chemical potential energy, directly proportional. What you'll notice is that we have never used in any of this the terms free or bound water. And the reason is because those are very confusing terms. They are not defined scientifically. They have different meanings in different situations or connotations. So there is a methodology within the pharmaceutical industry to measure bound water. It's a quantitative measure that you do based upon different temperatures and what the difference is in weights based on when you expose it to each of those temperatures. That simply is trying to get at that water that's more closely associated with these with solutes, but it doesn't have anything to do with energy. It's talking about a quantitative measure, where water activity is a qualitative measure of the energy. It's the relative energy of water overall within a system compared to what it would be if you were dealing with pure water. And that's why we have our equation here, again, related to the fugacity. Remember, that's escaping tendency. So the way we measure escaping tendency is by putting the sample in a sealed chamber and then allowing the water vapor molecules who have enough energy to be able to escape in the vapor phase and exert a pressure. And that's our partial pressure, or the vapor pressure of water, at a certain temperature, because that is influenced by temperature. And at that same temperature, if we divide that by what the vapor pressure would be if you were dealing with pure water, we get a ratio. The lower this vapor pressure, the smaller the water activity, the less the water in that product is behaving like pure water. So at a water activity of 0.50, it is not correct to say that 50% of that water is free and 50% is bound, because that doesn't really have any meaning associated with it. This is not a quantitative measure. What it does say is that the water in that product overall has 50% of the energy that pure water would have. And the reason why that's important is because it then provides us all the types of information we need about what water activity impacts. Water activity is also related to relative humidity. This is the same way that you measure relative humidity in the air, except that it's not in a state of equilibrium. So we say water activity is equal to the equilibrium relative humidity divided by 100. But that relationship is important when you start thinking about samples being exposed to room humidity, where those things are related. So in terms of how we measure water activity, so we've got our product. It could be food, could be pharmaceutical. We put it inside of a sealed container. Water molecules will escape into the vapor phase, and that again is being determined by what kind of interactions are going on in this product. Some water molecules whose energy has been transferred into those interactions will not be able to escape, and that's where that term bound comes from. So in terms of understanding what's happening with these water molecules and why they can't escape in the vapor phase, that can be useful. But again, we're not making a quantitative measure of amounts of bound water. We're just simply saying that binding actually influences the energy, which then changes the water activity. And you would compare that value to what you would have if you were to put in pure water at the same temperature. So, of course, water activity is not the same thing as moisture content. It sometimes gets confused. But in reality, moisture content is a quantitative measure of how much water is present, while water activity is a qualitative measure of the energy status of water. So it doesn't matter how much water is present. It matters whether or not what's happening with that water, what interactions is it going through, and what is the impact is that having on the energy of that water. So two different products can have the exact same moisture content, but have very different energies of water or water activities depending on what is in the product. And that's true even if you do this with a Carl Fisher moisture. It's still a quantitative measure, and is not the same thing as water activity. So what are the factors that affect water activity? So in very dilute solutions where you have a solute in a binary solution with water, only thing going on in those cases are what's called colligative effects. And it's just simply interactions between the solute's presence and the water that influences or impacts the probability that a water molecule can escape into the vapor phase. So these are happening but they are the only things happening in very dilute solutions. And so we say that those behave ideally. But that's very rare, and it's not something you would normally measure water activity on because it's going to be quite high. So what we're most interested in is the matrix effects, or these surface interactions. These are ionic interactions between things like salt and water, where there's a major transfer of energy during those interactions. Capillary effects, to a much lesser degree as well, impact what that energy is, but it really is a combination of all of these that determine what that final energy of water is in a product. So in terms of interactions that are happening, so when water molecules come into a product through adsorption or absorption, they are looking for anywhere that's polar, what we call binding sites. And when it finds those binding sites, of course, then it starts to have an interaction with the polar areas of the water molecule and the polar areas of whatever it is that's in there. And so right in that area where that interaction is happening, those water molecules, their energy is being transferred into that interaction. And so their energy is quite low as a result of that. And so their enthalpy will actually be high in comparison to that of pure water. But as they move away from those interactions, the outer layers of water, and this is why we use terms like monolayer, and then as you move to outer layers, those outer layers are having less influence from those interactions. They're actually are interacting with other water molecules, and as a consequence, there's not nearly as much energy transferred, and so their enthalpy actually starts to become the same as that of pure water. In which case, in our equation here, if that were to be true, then the water activity would be 1 if we took the exponential of 0. And so that's why the water activity of pure water is 1, just like we saw in Gibbs free energy equation. But as these interactions, and so then you can imagine the more of these molecules you have present for water to interact with, then the larger number of water molecules whose energy is going to be reduced, such that the overall effect is a lowering of water activity. Another thing that affects water activity is, of course, temperature. Water activity is temperature dependent. As temperature increases, generally the water activity goes up as well, but that's not always the case. It's very specific to each type of product. So you can see in this example, these are moisture absorption isotherms, and we'll refer to these several times today, but it's the moisture content plotted versus the water activity. So this is a description of how products take up and lose moisture. And what we can see here is at a given moisture level, so you're not changing the amount of moisture, you still can have changes in water activity. As the temperature goes up, the water activity is increasing, and as it goes down, it's decreasing. Why this is important is that this particular product, while safe at 25, has moved above that 0.7 level when it goes to 40, meaning that it's now susceptible to molding just because of a temperature increase. And it's possible to model this relationship between water activity and temperature through something called the Clausius-Clapeyron relationship, which basically tells us how to relate a change in water activity versus temperature for a given product. Now, this again is very product specific, but even using just a temperature controlled water activity meter, you can create and find out what the model is for that particular product simply by plotting the 3 points taken at 3 different temperatures. So you could take one at 20, 30, 40 degrees C, plot it, and that's all you need. Then you can, once you have that, the slope of that becomes our unknowns in this, which is basically the enthalpy divided by the gas constant. Once we have that value for this particular product, we can plug that into our equation and put in our known water activity and at a known temperature, and then predict what the water activity would be at a different temperature, such as 80 degrees C, where we put it in here in kelvins as opposed to a degree Celsius. But what we find out then is that while the water activity is 0.68 at 25 degrees C, we predict it will be 0.8 at 80 degrees C without having to have measured the water activity actually at that temperature. So this actually gives you the ability to predict what the water activity would be at any temperature, and it's amazingly good. We've tested this. There's publications available that I was part of where we looked at or design something to measure water activity at very high temperatures up to 80 C, and then compared to what the model predicted based on lower temperatures. And it's surprisingly good even up to very high temperatures in its ability to be able to predict that impact because it's thermodynamically linked. Now, in terms of types of water activity instruments, as I said, there are several different technologies listed in the USP 922. These uncommon methods wouldn't be listed. These are older ways of doing water activity, such as a hair hygrometer, freezing point depression, very rarely used anymore. So the main ones are the electrolytic resistive sensor, capacitance, chilled mirror, and then tunable diode laser. So let's spend a little time talking about each of these. First of all, the electrolytic resistive sensor is the type of sensor used in the instruments provided by Novacina and NewTek Group distributes those. And the advantages here, very stable. This sensor is within the chamber, is hidden away from the sample, so it's less susceptible to contamination than the other sensors because the sample can't actually get to it. As such, it doesn't require frequent cleaning. It's relatively insensitive to volatiles as long as you use filters. So chemical filters are available to protect that sensor. And so it allows you to read a very wide range of volatiles as long as those filters are placed in there. It has a very high accuracy level, certainly meets every stipulation. And in fact, USP 922 mentions this sensor by name. Measures the entire range. The only disadvantage would be that you can set the settings on this to be very stringent, which means when you use stringent requirements to end the test, remember this is an equilibrium test, and so you have to have achieved vapor equilibrium. And so the most important rule in terms of water activity test time is that the test time is dictated by the sample, not the instrument, because you have to go through this equilibrium process. And to make sure of that, you can set very stringent end of test requirements to indicate that change in water activity over time is very small, so that you for sure have gotten very close or achieved vapor equilibrium. There are, if you don't have those stringent settings, other systems, it's possible that you could get premature results because the settings aren't stringent enough, and it thinks or it meets the requirements of ending the test when in fact vapor equilibrium hasn't been achieved. And so you can certainly get tests, water activity tests, in 5 minutes, whatever time you want, as long as you set the settings less stringent. You just may not get very repeatable, accurate results. The other types of sensors are capacitance. This is the less expensive. That's its real advantage, is that you can make the cheapest instruments out of this technology. It uses a hygroscopic polymer between electrodes and measures the electrical change. Disadvantages here, high hysteresis, and this is a real problem. So every time you go up and down in terms of the water activity range, you risk having changes in the values, and they can be poisoned by volatiles. The chilled mirror has a high accuracy, can measure the entire range. Its biggest disadvantage is it can be affected by volatiles. There's no way to protect it from that. So you do have, if you have volatiles, you have to go to a different technology. It also is very susceptible to contamination on the mirror. If the mirror gets contamination, it will impact the reading. So it does need to be daily verified or more often, and requires frequent cleaning. Finally, the tunable diode laser. Its advantage is that it can be very short if you pre-equilibrate the samples. And so there are systems where you can pre-equilibrate within a container and then interrogate that headspace. That's very fast, but be clear that you have to put the sample in the container the night before and allow it to set for an extended amount of time within that container, and you can only use those containers. So you do have to prepare your samples ahead of time in order for that to work. There are other TDL instruments that don't use that approach. They work just like any other water activity meter, so they do require vapor equilibrium, so they're going to take the same time as any other methodology, because that's dictated by the sample. But they are not susceptible to volatiles, and that's the big advantage here. There's not any volatiles that they're susceptible to, so they can read all types of samples. Now, in terms of qualification of these water activity meters, there are guidelines within USP 922 to use saturated salt slurries. It also mentions unsaturated salt solutions. Saturated have the advantage of being reusable. It says in there, typically you're going to just use the acceptance criteria according to what the manufacturer provides or recommends. There are calculations in there for doing some determination of absolute error, but typically it says that use the manufacturer, and it's going to be by far more simple to do that than to try and use their calculations. And then, of course, temperature is a really critical thing. You would always have to report temperature with your water activity test, or it's meaningless. But you want to standardize. You want to use the same temperature, and that's really why you need instruments with temperature control to take water activity measurements. There are ones without temperature control that are offered, but not recommended because they can change throughout the year depending on what happens with the room they're in. The saturated salt standards make great standards because at its saturation, each salt has a different water activity, and they're reusable. So this is not a consumable. You can keep reusing these for up to 3 years. So you're not spending additional costs or ongoing costs on consumables to do those types of checks. Now, in terms of the document itself, it does talk about repeatability and reproducibility. It calls expanded precision reproducibility, but basically the difference here is this is when you take the readings all on from the same user in the same condition and laboratory versus when you take these readings by different users or on different days or in different laboratories. And it does give some restrictions or requirements for meeting certain levels depending on the water activity range. But keep in mind that the repeatability of the instrument is not done the same way as the repeatability of the sample. Those are two different things. The repeatability of the instrument you do with salt standards, and you just keep the sample in there and keep reading it without opening the chamber. If you want the repeatability of measuring water activity on a certain sample type, then you want to do obviously subsamples of that sample type, run again on each one, and then tracking the relative standard deviation across all those readings. For sample prep, main things here is to be consistent. In a lot of these cases, you have multi-component products like a gel cap, and you do have to cut that open to get to the interior. Otherwise, you only measure the water activity of the surface. And you do want to make sure you prevent moisture exchange. Back to this idea of water activity and relative humidity being the same thing. So if you expose your sample after you've taken it to the room, it's just changing to whatever room humidity is. So its water activity may be 0.3, but if you've got it exposed in a room that's 70% relative humidity, its water activity is changing to match that. So you do need to make sure they're protected, and keeping them in the cups with lids on it and only exposing it for a very short amount of time. And this is especially true of solid dosages, because they will change to room humidity very quickly because they're so hard on the surface, and they are better to crush those types of solid dosage tablets. You're better off crushing them than you are to try and read them whole. You'll get faster readings, and you'll get less variability because they aren't quite as susceptible in their crushed form to changing to the room humidity. Now, in terms of applications that are listed within USP 922, it talks about optimizing ingredients with water activity, making good excipient selections based on how they respond to water uptake and how they can control water activity. You want to prevent moisture exchange. You can use water activity to prevent moisture migration. Water activity impacts the degradation rate of active ingredients. Packaging needs to take into account the change in water activity that can occur due to exposure to different storage relative humidities. You can use it to monitor product safety and quality, provide microbial safety. And then, of course, still a justification for reduced microbial testing. So really the key to any kind of good drug release program to make sure you maintain quality and safety is to have water activity be part of it and identify what is the ideal water activity for your given pharmaceutical product based upon what the most likely modes of failure would be for that particular product. Now, in terms of optimizing ingredients and processing, again, the idea here is to identify what is that ideal water activity that's going to maximize shelf life, and then you process to it. That requires that you constantly check the water activity as a processing tool to make sure that you're in that right range or correct range, because if you under process to too high water activities, then you have the potential for having safety issues and breakdown problems. But if you overprocess, that's not good either. In a lot of cases, going to too low water activities can also result in problems, textural problems, different changes. But it also means that you're losing product. And so removing water through whatever processing you're using and going beyond what's needed is just wasted energy and wasted product. So it makes complete sense to identify what that ideal water activity is, and only process to that through checking water activity and not making any kinds of assumptions or trying to go off moisture content, because water activity is what is driving the control and the safety. Now, in terms of excipient selection, excipients are, of course, bulking agents. Their purposes vary depending on certain circumstances, but one of their main functions is to protect the active ingredient. And they can come in multiple forms. And it depends on what their role is in terms of what you would actually use. The most general category we can put them into is either crystalline or amorphous. And we won't spend a lot of time talking about the differences between those. There are sources for that or other webinars that I provide that talk more about this. But basically, the crystalline, very formed lattice has very distinct properties. Amorphous is in a glassy state and can experience glass transition. But some of the things to think about then are uptakes of waters of hydration, deliquescence, hygroscopicity, dissolution, and phase transitions. So if we take a look at amorphous versus crystalline, we can see those differences I was talking about very clearly through a moisture absorption isotherm. And you can see how different those moisture absorption properties are. Even though these are the same chemical, it's both sucrose, they behave very differently depending on if they're crystalline or amorphous. So obviously, you don't want to use a crystalline form of sucrose if you've made all your formulations based on it being amorphous. That would be a disaster. But in terms of whichever type you're using, if you're using crystalline, then the deliquescence point becomes your most important point for stability. Whereas if you're using amorphous, it's that glass transition that becomes your most important point. And both are induced through water activity. For instance, if you had a product or an excipient who was supposed to be anhydrous, if it takes up a water of hydration, that will impact its properties. It will change dissolution properties. And so if you're expecting it to be anhydrous, or if you're expecting it to have a water of hydration and that changes, that can be detrimental. And you can use these isotherms and identify exactly what water activity would level you would have to go past in order to either take on a water of hydration or lose a water of hydration. In addition, you can very clearly find the deliquescence point. Deliquescence is induced by water activity. In fact, it's why saturated salt slurries work. Basically, a salt slurry, when it goes into when you put it in a slurry form in saturation, it's basically on that line that goes vertical. And that's why you can, even if you change the amount of moisture, you still maintain the same water activity. But the deliquescent points becomes very important for any kind of excipient in a crystalline form. And as you get closer to this point, you start to have surface capillary condensation that results in stickiness. And so you want to identify where that is and make sure that the product remains away from it. Now, in terms of hygroscopicity, that can be important depending on the range that you're looking at. So you can see these different excipients all behave very differently depending on the water activity range. Some of them at very high will become extremely hygroscopic, and the steeper the slope of the curve, the more hygroscopic the material is. That also means that steep curve that you can take up moisture in these excipients without having a subsequent big change in water activity. And that can be important in terms of active ingredients because you're maintaining the water activity of the active ingredient, even though water is being absorbed, maybe because of exposure to high humidity. And then, of course, dissolution is something that millions of dollars are spent on trying to determine these dissolution properties, making sure that the efficacy of the active ingredient is delivered at the right point. And so tests are done to figure this out. The problem, though, and that's fine, but then you go forward with an assumption of a certain dissolution properties for a product. And if that changes because of changes in the material, like deliquescence or like phase transitions, that will all impact these dissolution properties. So even though it had a certain dissolution property at the time when it was processed, if it goes through any kind of changes after that due to changes in water activity, then those dissolution properties will have changed. And if we look at that from an amorphous standpoint, again, these are glassy powders usually down in as this glassy state. They have a metastable state there where their shelf life is going to be quite long. They tend to perform quite well. The problem is if these take up water, and if they do absorb water, their water activity is going to increase, and it will match this isotherm. And at some point, it will reach a critical water activity where it will experience that phase transition, and it will go transfer to the rubbery state. And then it starts to cake and clump. All problems start to speed up. Shelf life goes way down once you pass that critical water activity. So for these types of excipients, it's very important to know where that point is so that you can make sure they're kept at water activities lower than that. And this value will change with temperature. As temperature goes up, it moves to the left. So you have to make sure you're low enough to be below that, even if you were to have some type of temperature abuse. Now, in terms of moisture migration, this is also controlled by water activity. Water moves from high energy to low energy. Remember, water activity tells us about energy. Water doesn't move from high concentration to low. And so if you put two materials together like a gel cap and a excipient powder with an active ingredient, and that powder's at a much different water activity than the gel cap, you're going to have moisture migration. Depending on which one's higher and which one's lower, it always moves from lower to high, but you'll come to some equilibrium point, which is not the midpoint between those two values. It's dictated by the absorption properties of each one of these materials. And so in that process, depending on where that equilibrium water activity is, there's always a chance that these caps are going to shrink, or they're going to become sticky, or that you have increased degradation of the active ingredient. So the key there is to try and manufacture the interior and the outer portion to the same water activity as much as possible. If you can't, and you do have to put them together at different water activities, you can calculate that equilibrium water activity through modeling techniques. And that's what's being shown here. I don't provide the model itself, but if that were something you're interested in, I certainly would be able to help you with that. But basically, through this work, we could predict that the final water activity would be 0.68, when in reality it was 617, 634. So very, very close in terms of the prediction, just based on the modeling work. And then we can also very closely predict what the final moisture content will be of each of those components. Once that's known, then you can make a determination that if at that water activity, are you going to have problems? Finally, the chemical degradation of the API is, of course, a very detrimental mode of failure, because you've essentially gotten rid of the efficacy of the drug itself. So if it can't deliver the active ingredient, then it's useless. These active ingredients can be very sensitive to things like hydrolysis or enzymatic reactions. In general, as water activity goes up, reaction rates increase. The rate at which they increase will be specific to the matrix, the active ingredient, and the reaction. But in general, we can say that reaction rates increase as water activity increases. Now, to figure out exactly by how much does that increase happen, it's possible to do some studies that allow you to be able to look at this in an accelerated form, because a lot of times it's hard to wait the full time to see what kind of degradation is going to occur on these active ingredients, because especially if we're talking about years of shelf life. And so there's a desire to be able to accelerate that. And so accelerated stability testing is something that's gained a lot of popularity within the pharmaceutical industry. And basically the idea here is that you accelerate those storage conditions to try and speed up whatever bad is going to happen. And if you track the rate of that at accelerated conditions, you can extrapolate back to normal conditions and make a prediction of what that rate of reaction is going to be, which ultimately tells you your shelf life. And so it's possible to do that, but it should include temperature and water activity. In a lot of cases, maybe it only includes temperature, but you have to include both. And you collect that time series data, figure out what the rate constant is of the reaction. And then it's possible, if you do that at several, say 3 temperatures, 3 water activities, to come up with a model that will actually predict what the rate would be given any water activity and temperature. And this is called hygrothermal time. There are other models, or at least one other model out there that does both water activity and temperature. However, it's a little different than this and not derived from fundamental principles, whereas this one is derived from the Eyring equation for rate plus Gibbs free energy. So it is a very good model. It's been published as far as being able to do a good job of predicting rates of reaction. If you're interested in learning more about doing this type of work, again, I can provide additional information if you want to reach out and contact me. And then finally, package optimization. So all of these products are, of course, are going into packaging, and the packaging purpose, among many other things, one of them is to prevent moisture exchange. So that if your pills sitting in these pill bottles are exposed to high humidity, which in a lot of cases they are, think about a bathroom with a shower on, then it's important to understand what are the moisture permeability properties. Because thinking back to what we've talked about, there's these critical water activities where and an ideal water activity for these products. And so you want to keep it there. Well, if that water activity is 0.3, but it's in this bathroom that's frequently getting up to 75, 80, 85% relative humidity, there's always a chance for moisture penetration through the packaging, changing the water activity in the interior. And so it's important to know how much change is possible. If you know the water vapor transmission rate of the pill, of the container, you can make calculations about how much the water activity is going to change of your pharmaceutical product given a certain set of conditions. We do that through a fixed first law. We can also predict if you have a certain product and you want to know how to make it never get past a certain water activity, we can also reverse that modeling and do calculations about what the actual water vapor transmission rate has to be. But what you'll notice is this in action, which is that if you go into countries where the relative humidity tends to be high year round, almost all pharmaceutical solid, low water activity products are packaged in blister packs. And that's because they have better permeability properties. More expensive to do that, but it gives it that added protection. So again, if you would like some help with trying to look at packaging and do modeling about water activity change in package, happy to help with that as well. And then finally, we talk about microbial safety. And really, this is what water activity is known for, but it provides the same safety and protection in pharmaceuticals as any other type of product. The rules still stay the same and apply. And that is that once you're below 0.7, that's the practical limit for all microorganisms. Below 0.86 is where your pathogenic bacteria stop. Now, why is this? And this is, again, related back to our definition for water activity being the energy of water. This is why it impacts microbial growth. Not because water activity is the water that's available for microorganisms to grow. That's not a correct definition, but because of the energy relationship. So a microorganism has a certain internal water activity because the energy is the water in there has a certain energy. And when it encounters an environment where the water activity is lower, and so the energy of the water is lower in the environment, water moves preferentially out of the cell from high energy to low energy. And when it does that, it loses turgor pressure and it can't go through its metabolic activities. The organism has an ability to respond, and each organism has its own unique ability to respond to what we call osmotic stress, these low water activity conditions. Tries to lower its interior water activity in an attempt to try and get lower than its environment, so water will start to move in again. If it can do that, then it will start to grow. If it can't, then it has to stop. It's forced to go dormant. And these organisms each have their own kind of unique way of going dormant. Some can form spores, and they can last for a very long time. They aren't going to actively grow at these low water activities, but they're still there, and they're still viable. And if they get into water activities that are high at some point during the life of that product, it will start to grow again and can result in contamination. So water activity is not a sterile step. It's not a kill step. It doesn't eliminate microorganisms. It simply prevents their proliferation. And so here are some of those limits for the pathogenic bacteria. Each organism, again, has its own unique point where it stops growing. Keep in mind that these are all under optimal conditions for everything else but water activity. So water or microbial growth, of course, is impacted by things beyond just, and factors beyond just water activity, including pH, temperature, and many others. And those combined can actually influence microbial growth and cause it to stop at water activities higher than this through the influence of other factors or growth factors. But these values represent absolute minimums. So everything else was perfect for the growth of the organism. The only thing that's limiting it would be the water activity. And so they will not grow below this. Now, they may stop growing at values higher than this water activity if that water activity is combined with something like pH. And we call that hurdle effects. Now, in terms of water activity measurement, of course, as we said earlier, NewTek represents Novacina, and they're the makers of the LabMaster Neo. And this is really the ideal water activity instrument for the pharmaceutical industry for several different reasons. It uses this resistive electrolytic sensor, very well understood, very stable, hidden away from the sample itself, provides the highest accuracy at plus or minus 0.003, very low maintenance requirements, as we indicated, protected from the chamber, can measure volatiles if you put filters in there. But it's also fully 21 part 11 compliant, complete with the audit trail that you can download. So this is really the only water activity instrument out there that meets full 21 part 11 compliant by itself, not in combination with the software program. And in fact, the audit trail is shown right on the screen. You can access at any time. Very high range in temperature control from 0 to 60 C. You can the end of test settings, as I alluded to earlier, where you can get premature results. In this instrument, you can set those. Those are user set. And so you can change them to be more stringent to make sure you're getting good results, or less stringent if you want to try and get faster results. But you'll know, because of this capability, exactly what you're doing in terms of accuracy changes by having a test that's faster. And that's really the beauty of the system. And in fact, it graphs it right on the screen, this equilibration process. Of course, it uses reusable salt slurries, so those your salt standards aren't a consumable. It also has automated verification and calibration. You take your salt slurry and you put it over the top of this point in the instrument, and it detects what saturated salt it is. It comes up on the screen, says, do you want to verify / calibrate? You say yes, you put it in, you close the lid, automates everything, comes up at the end and tells you what it did. So very easy as far as maintenance, very easy as far as calibration and maintaining the instrument. And really that all makes it the ideal instrument for the pharmaceutical industry. So if you're interested in learning more about USP 922 and the solutions that NewTek offers that meets those requirements, we would encourage you to contact us. In addition, if you're interested in trying to do any of the modeling that I discussed as far as applications, we also do offer consulting services where you can basically have me help you for a certain amount of time, work on any of this, and we have different packages that we can offer for those consulting services. And with that, I'll turn it back to Orna.
Well, thank you, Brady. Always a wealth of information. If you have any questions, please jot them down in the chat box. And as Brady said, he's available to answer some questions offline. If you want to discuss your specific applications or needs, feel free to reach out to us. We're going to be posting a survey in the chat box. I see it's posted already. Please take a few moments, not even minutes, to tell us, what you thought about the presentation. If you have any questions, if you'd like us to reach out to you. Brady, there's a question here about excipients. What type of excipient is the best for controlling water activity?
So again, it depends on kind of the situation that you're in, and the range of the natural range of the water activity of the product. But basically, the more hygroscopic the excipient is, the better it is at being able to take and handle water absorption without having subsequent big changes in water activity. And that's critical, again, because the excipient's basically protecting that active ingredient. So whatever the water activity of the excipient is, is going to be what the water activity of the active ingredient is. And so it's those sorption properties that become critical. And we do offer a solution for being able to generate those moisture sorption isotherm curves with our NEO instrument that I showed there at the end. There is a sorption isotherm kit that can be purchased with that NEO, where you can actually turn the instrument into a moisture sorption isotherm generator using the salt standards that you use to check the instrument. So it is possible to investigate those sorption properties of excipients using a NEO, and it's also a water activity instrument. So that's a nice capability for that NEO, and only the NEO is able to do that.
Brady, another question is asking about the typical analysis time for dry powder samples. What is the typical analysis time?
So yes, and again, this is another very good question. Thinking back in terms of remembering that the sample what dictates the test time, not the instrument. And so with powders, especially glassy powders, they can actually, some of them can take quite a while to fully equilibrate, and even up to 20 to 30 minutes to get full equilibration. A lot of instruments, other instruments that measure powders will give results in a fairly short amount of time because the change is quite slow but significant. And so it's one product where the NEO really shows itself, because you can set the settings to the very least stringent you want and get results after, say, 5 minutes. But with this system, as long as you leave that sample in the instrument, it will continue to track water activity over time. It'll give you the value it got based on your stability settings, but then you can watch it and see exactly by how much that water activity changes if you were to leave it in there for 40 minutes. And that allows you to actually figure out what is the real equilibration time for that powder or whatever you're reading, and how much different was that from what I got after 5 minutes. If the difference isn't big, then you're probably okay reading it after 5 minutes. If it is big, then you really have to consider, is it worth trying to get such fast results when you're not actually getting correct results?
Thank you. Thank you so much, everyone, for joining us. And thank you, of course, Brady, for all this information. You have our information here on the slides. You're welcome to visit our website. We're accessible anyway. And we look forward to seeing you in future webinars. Thank you, and enjoy the rest of your afternoon.